Here with Dr. Michael Young from the Scapin's Eye Institute for the Innovations in Vision Restoration series. And so we're happy to have you here with us.
Happy to be here.
And so maybe you can tell us a little bit about your background, the work that you've done maybe at the Scapin's Eye Institute, and maybe any foundational background before that.
So I'm from Western Pennsylvania. I grew up in Butler, just north of Pittsburgh. Went to Pittsburgh for my undergraduate, and then went to Cambridge in the UK for my PhD. Did a postdoc at MIT, and then I've been at Harvard for about 10 years now, 12 years now, doing mostly retinal progenitor cell work, mostly work trying to regenerate the retina, try to grow new stem cells that can replace lost cells.
And I understand you have sort of pioneered the area of retinal stem cells and isolation of retinal stem cells.
Yeah, we've been successful in isolating stem cells from the eye of many different species, including humans. Mm-hmm. So we now have human retinal stem cells that we are now testing in pig models of retinal degeneration to try to see if we can restore vision in blinded animals first, which is a necessary prerequisite to clinical translation.
Okay. So what sort of diseases would you target with these retinal degenerative
Initially,
cells?
we're targeting diseases like retinitis pigmentosa. Eventually, we think this will have some applicability to diabetic retinopathy and to macular degeneration as well. But right now, I think the first order disease for us to target is retinitis pigmentosa.
Okay, sort of an initial target. And then would this be applicable then for age-related macular degeneration,
Certainly,
a fairly common
certainly.
pathology?
Yeah, I think initially there'll be some work replacing the retinal pigment epithelium in macular degeneration, but I think we all know that's not going to be enough. And so in diseases like macular degeneration where photoreceptors are lost, you're going to have to replace them if you want to restore vision in patients. So I think initially there probably will be some pigment epithelial trials, and that's going to happen fairly soon. And then soon after that, we think we'll be able to begin a trial transplanting stem cells for photoreceptor regeneration.
Okay. And the idea is that because these cells are lost, you've got to physically put a new cell in there to replace that function.
Exactly. So in frogs and fish, they can regenerate a retina that's damaged. We've lost that ability. For whatever reason, mammals have lost that ability. So mice, who we frequently use as study organisms, don't regenerate their retina, and people don't either.
Right. Maybe we can switch to the subject of motivation. What motivates you to do this type of research? Why is it that you chose this area and this particular interest?
It's interesting. I think initially I was a neuroscientist, so I came to Pitt wanting to be a neuroscientist. Got a degree in behavioral neuroscience. But early on I met up with a guy at Scaife Hall by the name of Ray Lund, who was the chairman of neurobiology at the time, who was doing some really freaky transplants of retina into brain.
Oh, wow.
As an undergraduate, that just lit my imagination up.
Right.
And so I initially started studying the retinal transplant as a means to study development and plasticity in the central nervous system. And then when I was at MIT, a position opened up at Harvard to study retinal transplantation itself.
Right.
And I decided at that point to sort of go back into that field, leaving basic developmental questions aside for a while and focus more on the translational side of restoring vision in patients. And that's sort of been what's been my goal for the past 10 years or so.
Okay. And ongoing.
No shortage of things to do.
Absolutely. Well, thank you for being with us, Michael. We appreciate
Pleasure to come
it.
back to Pittsburgh and see some old friends and meet some new people.
Very interesting work.
Thank you.
Thank you.